So the ammonium here, freed by glutaminase in the liver, readily enters the mitochondria, where it can enter into the synthetic pathway of urea synthesis. So a functioning urea cycle is essential for the disposal of nitrogen from catabolic processes. And if dysfunction occurs, the accumulation of ammonia can be life-threatening. So you'll notice that the urea cycle, which occurs in the liver, spans both the mitochondrial and the cytosolic compartments. Initially, the free ammonium diffuses into the mitochondria and is combined with carbon dioxide to generate our initial product, which is carbamyl phosphate. What's important to notice here is that the synthesis of urea is an anabolic process and requires ATP. So in the fed state, the ATP is coming from the oxidation of dietary substrates, whereas in the fasted state, this ATP is coming from the primary process of beta oxidation in the liver. So from here, carbamyl phosphate condenses with ornithine and is shuttled out of the mitochondria as citrulline. From here, we'll add our second amino group from aspartate in a reaction with arginosuccinate synthase. Again, this also requires ATP to synthesize arginosuccinate. Arginosuccinate is then cleaved into fumarate and arginine by arginosuccinate lyase. You'll note that arginine is a non-essential amino acid as it is synthesized in the urea cycle. And finally, arginase is responsible for cleaving urea from arginine, and our remaining ornithine is transported back into the mitochondria. So urea is a compound that contains 2 amino groups and is then sent to the kidney to be incorporated into urine. But keep in mind, the synthesis of urea is in the liver. Our pathway is regulated at one key step, and that's at the enzyme carbamyl phosphate synthetase 1. This enzyme requires the allosteric activator N-acetylglutamate. It's interesting to note that the synthesis of N-acetylglutamate is enhanced by arginine. So as arginine is an intermediate of the urea cycle, as it elevates, it will also stimulate the production of our allosteric activator. Therefore, the cycle provides positive feedback on itself. Again, here the regulation of carbamyl phosphate synthetase is our most important point here, and it requires N-acetylglutamate as an allosteric activator. So again, what's important to note about our urea cycle is, unlike other pathways discussed, the urea cycle functions independent of hormonal control, as it functions to dispose of nitrogen, either from excess dietary sources or from protein catabolism or turnover. In the fasted state, keep in mind this is especially important as we require carbon skeletons from amino acid catabolism for the process of gluconeogenesis. So now let's finish up with some clinical scenarios which will help us identify different deficiencies along this pathway. So we'll look at the metabolic panels of 3 different infants, presumably born with deficiencies in the urea cycle, and that would be indicated by this high level of ammonia. So if ammonia is elevating, it would suggest that there's a deficiency in the ability to dispose of ammonia through the urea cycle. And we'll use these other 3 values, urine orotate, blood citrulline, and blood arginine, to help us determine what enzymatic deficiency these individuals have. So let's first look at infant 1, and let's stop for a moment and talk about urine orotate. So urine orotate comes from an alternative synthetic pathway that accepts carbamyl phosphate. So what we'll see in our final week is that carbamyl phosphate is also synthesized by a cytosolic enzyme, carbamyl phosphate synthetase 2. And carbamyl phosphate is subsequently used in pyrimidine synthesis, and one of those intermediates of pyrimidine synthesis is orotate. So given that there's two metabolic pathways that can accept carbamyl phosphate, what happens is in deficiencies of the urea cycle that cause an accumulation of carbamyl phosphate, the carbamyl phosphate can then enter into pyrimidine synthesis and be used in the synthesis of orotate. So in this case, urine orotate is low, which would suggest we are not accumulating carbamyl phosphate. So this intermediate is low. We're also noting that blood citrulline is low, as well as arginine. So given that the products here are low in the majority of the pathway, this would suggest that only one upstream enzyme could be deficient. These would all suggest a deficiency of carbamyl phosphate synthetase. So if this enzyme was deficient, it would reduce synthesis of carbamyl phosphate. The reduction in carbamyl phosphate would then decrease citrulline pools as well as arginine pools. Again, our ammonia would be high because in the absence of this enzyme, we would accumulate free ammonium. And this is in fact the correct answer here. We have a deficiency of carbamyl phosphate, and these are the diagnostic values that would be consistent with this deficiency. So let's look at the second infant. Again, urine orotate, which comes from an alternative pathway of carbamyl phosphate metabolism, is not existent or low. This would suggest that we're not accumulating a carbamyl phosphate. We also know that we're producing citrulline at very high levels, and blood arginine is low. And again, our blood nitrogen is high. So if we have an accumulation of citrulline here, and this would suggest that there's a deficiency in either arginosuccinate synthetase or arginosuccinate lyase. A deficiency in either of these enzymes would result in elevated citrulline and a decreased production in blood arginine. This is in fact correct, as the enzyme deficiency represented here is in either of these two enzymes. In this case, it's more likely that arginosuccinate synthetase is deficient due to this extreme level of citrulline elevation. And finally, let's look at this last scenario. And in this scenario, we have an elevated urine orotate, which would suggest an increased production or increased accumulation of carbamyl phosphate. We also know that citrulline is low, as well as arginine, and again, ammonia is high. So given that we have an increased circulation of ammonia and an increased amount of carbamyl phosphate, this would suggest that carbamyl phosphate synthetase is active in producing carbamyl phosphate, but that our ornithine transcarbamylase is deficient, as we're not using this carbamyl phosphate pool to synthesize citrulline. This is in fact the case. This would be a characteristic of an ornithine transcarbamylase deficiency where carbamyl phosphate elevates and is used in pyrimidine synthesis in the cytosol, resulting in this high level of orotate, reduced levels of citrulline, and reduced levels of arginine. So this finishes our discussion of the urea cycle, and use these three examples to help you better understand the pathway. Keep in mind there's only one regulatory step, and that's at carbamyl phosphate synthetase. So this helps simplify the pathway.